Bismillah ar-Rahman ar-Rahim, in the name of Allah, the most gracious and the most merciful. We start another paper 4.3, video 2, which is question 4, 5 and 6. Question number 4.1, cause of genetic disease severe combined immunodeficiency SCID is a mutation in the ADA gene. This mutation results in a deficiency of the enzyme ADA, adenosine deaminase. Although ADA is found throughout the body, it is especially active in lymphocytes. The absence of functional ADA causes the buildup of toxic metabolites that kill lymphocytes and damage organs. Babies are often diagnosed with SCID by 6 months old. Treatment can generally improve the life expectancy of children with SCID. Some treatment options are available. Enzyme replacement therapy with recombinant human ADA made by genetically modified GM E. coli. Weekly intramuscular injections are given. Weekly. Bone marrow transplant is a well-matched donor such as a close relative can be found. And the third is gene therapy. Suggest and explain why it be more appropriate to use enzyme replacement therapy to treat SCID instead of a bone marrow transplant. Why? Donor not needed. You see, in enzyme replacement therapy, you don't need a donor. As immediate effect, easy to administer the treatment, no immune response, less invasive. It is cheaper and quicker and lesser risk to health. Donor will have to have a lot of tissue matching and all that kahani, and that may or may not take place, may not be possible. So donor not needed, immediate effect, easy to administer, no immune response or story of rejection, tissue rejection, less invasive and cheaper and quicker. Then coming to the B part of the question, outline the procedure used for gene therapy treatment of a person with SCID. First, you give them a drug to increase the number of stem cells in the bone marrow. Then you take a virus, which is a vector which will contain the normal healthy allele. Then you remove the stem cells and you mix the stem cells with this viral vector so that the normal allele can be transferred. And then you give radiotherapy, drugs to kill the stem cells in the bone marrow. And then the stem cells that you have transduced or changed, then you infuse those into the blood. And the lymphocytes will produce the functioning ADA. So it's a very long story. It's not something very easy. First, you give a drug to increase the number of stem cells in the bone marrow. Then you take a virus or a vector which contains the normal allele. Then you remove the bone marrow or the stem cells. Then you mix the stem cells with the viral vector. Then you kill the stem cells in the bone marrow because you want to make some space. And then you give an infusion, like an IV infusion into the blood. You inject these the ones which you have changed, the stem cells. And now these new lymphocytes will produce the functioning ADA. So now you can see how I'm going to word it. Give the drug to increase stem cells, then take a virus with a healthy allele, then remove the stem cells, then mix the stem cell with a viral vector, then radiotherapy to kill the stem cells. Why? We want some more space. And then you transduce stem cells will be injected into the blood, and the lymphocytes will produce the functioning ADA, which was not being produced earlier on. Then part C, this is a very favorite question, suggests the social and ethical implications of gene therapy for SCID that need to be considered before treatment is carried out. It's very expensive. It's a long-term treatment. No longer the person is chronically ill, maybe a better quality of life, no need for regular injections. Then there could be cultural or religious objections. Then no donor is needed. More money is available to the health system to other people in the long term. But then we don't know what this will do. So it may cause cancer. This whole treatment could be very stressful. So social and ethical implications. Ethical, of course, means ethics, means cultural and religious objections. People might say, okay, well, we really don't know. I mean, what is going to be the effects of this, and is it religiously allowed? Can we tamper with the genome of a person? What would happen 10 years from now? What would happen 20 years from now? Does it affect his health in any other way? With the people with this, can they ultimately start suffering from cancer or not? So that is why all these points have to be considered. And these are sort of some points which you can write in every question which says social and ethical implications. So a lot of points, but it's only a 3 mark question, so you only have to give me 3 points. It's expensive. It is, of course, it's better quality of life because the person doesn't have to have regular treatments. You don't need regular injections, only a single treatment is needed. Then there's cultural and religious objections. Then no donor needed, may cause cancer in the long term, and of course, it is very, very stressful. Question number 5. A freshwater fish species Oryzis latipus has individuals with 4 body color patterns as shown in table 5.1. So we have a red one, we have a white one, we have a red with black spots, and we have a white with black spots. So we have 4 body color patterns. 2 unlinked genes determine the body color pattern shown in table 5.1. One gene controls whether body color is red or white, dominant R, small r, white. The other gene controls whether black spots are present or not present. So dominant B with black spots and recessive B is without black spots. Right? So a fish that is homozygous recessive at both loci is white. Why? Because this is white and this is going to be without black spots. So it's going to be white. Genetic crosses were carried out to investigate the inheritance of the 4 different body color patterns. Males that were red with black spots, males, and homozygous at both loci. What does that mean? Black spots, red is dominant. So red had to be big R, big R. And black spots is big B. So big B, big B. Homozygous at both loci, understand that English, were crossed with females that were white. So R, 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 R. And were white. So the white means they did not have any spots. So they would be small b, without black spots and white. So the F1 offsprings were all red with black spots. Why? They were all this and this. So they were all red with black spots. Big R, small r would be red, and black spots could be big B, or big B, small b as well. The F sprongs were then crossed to produce the F2 generation. So that means you cross these with this. Were crossed to produce the F2 generation. A table 5.2 shows the observed numbers obtained for each of the 4 different phenotypes for the F2 generation. Table 5.2 compares the observed numbers and the numbers that would be expected to the F2 generation for a normal dihybrid ratio. Calculate x square for the F2 generation. The formula is this. Now, how do you really calculate it? Expected is 9:3:1 ratio. 9 ratio 3 ratio 3 ratio 1. So 279 were observed, 95, 96, and 30. Now you add up all these and then you calculate the 9 out of 16, then 3 out of 16, and 1 out of 16. So they've actually given you the expected ones and they've done that for you. Now you have to do O minus E. You have to do this. You had mostly it's all been done for you. So this would be minus 2.25, and O minus E whole square would be 5.0625. Then O minus E whole square over E will be 0.018. You must see it's in 3 decimal figures, so you all must do that. And then the white one, 30, 31 point O minus E will be minus 1.25. And then this was O minus E whole square would be 1.5625. And then this would be 0.050. And this, of course, x square would be 0.139. 13, my pen doesn't seem to work here. 0.139. Or you could have said 0.14. That is also correct. So 3 marks. So there is, how do you get the marks for this? 1 mark for each column. So you got this column right, 1 mark, second column right, 1 mark, and the third column right, 1 mark. 1 mark for each column. So you got that right. Well, you got the 3 marks. Then it says the critical value at probability 0.05 and 3 degrees of freedom is 7.815. Now your value 0.14 is much less than that. Comment on whether the null hypothesis should be accepted or rejected. When your value is less than the critical value, what do? X square value is less than the critical value, which was 7.815. So the observed numbers are not significantly different to the expected numbers. Any differences are due to chance. That's a favorite way that how we write this, and everybody must know that X square value of 0.14 is less than 7.815. So 0.14 is less than 7.815. This is the 7.815 and this is the 0.14. So it is less than the critical value. So the observed numbers are not significantly different. Not significantly different to the expected numbers. And any difference due to chance. Well, the null hypothesis, when the value is less than the null hypothesis, is accepted, which means this. The null hypothesis, that doesn't get you any marks if you said accept the null hypothesis. That doesn't give you any mark because it doesn't give you an explanation of that sentence, accept the null hypothesis. Accept the null hypothesis means that there is the only difference is due to chance. So there is no significant difference between the observed and the expected numbers. If you said accept the null hypothesis, I'm sorry, you won't get any marks because we want an explanation of that. What does it mean, accept the null hypothesis? Then coming on to another part of the question, further analysis of the results from the F2 generation table 5.2 show that there were no white males or white males with black spots. In O. latipus, females have the X chromosome and males have an X and a Y chromosome. It was deduced on O. latipus the gene that controls body chromosome is located on the X and the Y chromosome. The gene then controls whether black spots are present or not is located on an autosome. So this is some information they've given you, and now they say to produce the F2 generation, red males with black spots XR, YR, BB were crossed with red females and black spot XR, XR, BB. Complete the Punnett square to show the genotype and phenotypes of the F2 generation. Use the symbols this, and use the symbols B and B for the alleles of the gene that control whether black or spots are present or not. Sum of figure 5.1 has been completed for you. I mean, they made it easy for you, so you just had to complete this table. So as you can see, I've completed this. So four boxes that you have to complete and 4 marks. So you could have got the 4 marks if you had got this all correct. We had to know XY was a male and XX was a female, and that was easy to deduce, not a very difficult part of the question. Then D part of the question, explain why there are no white males or males that are white with black spots in the F2 generation. Why? Because the allele R is on the Y chromosome. So all males inherit the dominant red allele, and only Y capital R is present in the gametes. No small r is on the Y chromosomes. So males never inherit the recessive white allele. Allele capital R is on the Y chromosome. All males inherited the allele capital R. No allele small r on the Y chromosome. Males never inherit the allele small r. Then part E of the question. In another cross, red males with the genotype X chota r X Y bada R BB were mated with white females with the genotype XR XR BB. All the male offspring were expected to be red and all the female expected to be white. The observed results show that the offspring included two red females out of 253 and one white male out of 198. Suggest an explanation for this unexpected result. Very simple. Unexpected means it's a mutation. That means that's a Chang'ar thing. So mutation, 1 mark, then crossing over, then you could given me the detail of the mutation. It could be a substitution, or it could be a substitution, or it could be addition or deletion. Then crossing over, all the dominant red allele from a Y chromosome was passed on to an X chromosome. So that is, of course, detail of the mutation. And of course, if you had said any of the other point, that the R allele, the capital R, from a Y chromosome to an X chromosome. So this is actually related with crossing over. Crossing over could have resulted in the capital R allele from a Y chromosome going on to an X chromosome. And if you know the details of crossing over, then you would know it. So mutation and detail of mutation, 2 marks, and crossing over and this thing, another 2 marks. So 4 marks, and you could have given me any two out of these. Then question number 6. Figure 6.1 shows part of the Bowman's capsule of a kidney nephron, lumen of the capillary A, B, C, not to scale, lumen of the Bowman's capsule, name structures A, B, and C. A is the endothelial cell. I mean, I give you a lot of examples in class, and I give you a lot of endothelial cell, or just, sorry, not cells, but just cell. So endothelial cell got you 1 mark. Then B was the basement membrane. Very favorite question. They keep on asking you this again and again. If you do the past, C was the podocyte. Then, of course, we come to a slightly bit of maths. Fluid is forced into the Bowman's capsule by ultrafiltration to form the glomerular filtrate. Describe the role of structure B in ultrafiltration. Structure B is the basement membrane. What is the main thing? Acts as a filter, prevents molecules with more than 68,000 mm from passing through, stops large plasma proteins and red blood cells to pass through. So acts as a filter, prevents molecules more than 68,000 mm from passing through, and stops large plasma proteins and even red blood cells. So you could have even said the RBCs of the red blood cells. Now coming to the C part, which is again a little bit of maths. The main component of the glomerular filtrate formed in the Bowman's capsule is water. Most of these water is reabsorbed as the filtrate passes along the nephron. 180 dm³ of water forms part of the glomerular filtrate each day. 1.4 dm³ of water leaves the body in the urine each day. Calculate the percentage of water in the glomerular filtrate that is reabsorbed as the glomerular filtrate passes along the nephron. Show your working and give your answer to one decimal place. Now, what was the working? 180 minus 1.4 or minus 1.4 divided by 180 into 100. A favorite question of theirs. And this is 99.2. And of course the unit is written here. So 180 minus 1.4 over 180, or you could have said 178.6 over 80. Into 100. This of course gives you 99.2%. Now coming to the D part of the question, which is again a very direct question. Describe how the action of ADH on the cells of the collecting duct leads to the reabsorption of water in the glomerular filtrate. And this is for 7 marks, and that will finish this question. That's a very simple basic question. ADH acts as a cell signaling molecule. ADH binds to receptor on cell membrane of collecting duct cells. Second messenger produced, which is cyclic AMP, then enzyme cascade, then vesicles or aquaporins activated. The aquaporins or the vesicles moves towards the cell membrane. Aquaporin added to cell membrane increases cell membrane permeability to water. Water moves out of the collecting duct by osmosis and into the collecting duct cells or the tissue fluid and finally into the blood. Now, as you can memorize the points, ADH acts as a cell signaling molecule. First point, ADH binds to receptors on cell membrane of collecting duct cells. CAMP second messenger produced. Enzyme cascade. Aquaporins activated. So I'm just going to use the words which I want to talk about. I like using these words. Aquaporins activated. Vesicles with aquaporin moves towards the cell surface membrane. This is the collecting duct, and you have these are the cells. And the aquaporins, of course, come and get attached here, and they were inside previously. And they come and get attached here. And then, of course, all the water from this is going to come out here into the cell and finally into the blood. So vesicles with aquaporin move towards the cell membrane. Aquaporins added to the cell membrane increases cell membrane permeability to water. Water moves out of the collecting duct by osmosis and into the blood. Ultimately, finally, it goes into the blood. So the water potential of the blood is maintained. The blood was too concentrated. Now the blood is going to normalize and it's not going to be too concentrated. Then coming to question number 7, sarcomeres are the functional units of the myofibrils or the muscle fibers of striated muscle. The arrival of an action potential at the sarcoplasmic reticulum of a striated muscle fiber can lead to the shortening of a sarcomere. The shortening occurs by the sliding filament model. Outline the sequence of events occurring after the stimulation of the sarcoplasmic reticulum that results in the shortening of the now that's again a very simple, I mean it's a lot of memory that you have to do, nothing very important. Voltage gated calcium channels, calcium ions leave the sarcoplasmic reticulum, calcium binds to troponin, troponin changes shape, exposing binding site on actin, myosin heads bind to this site, or you can say forms cross bridges, and myosin head tilts and pulls the actin, so which of course shortens the sarcomere. So this was very simple direct question. You either knew it or you just didn't know it. Now as you can see here, the above points which I've written in yellow, you could only have given me 3 of those. Because it says outline the sequence of events occurring after stimulation of the sarcoplasmic reticulum that result in the shortening of a sarcomere. So these were the 2 essential mark scheme points, and any 3 out of these then you could have given me. So that is why this is a 4 marks question. So open calcium channels in sarcoplasmic reticulum, calcium ions bind to troponin, troponin changes shape, exposes binding site on actin, myosin head forms cross bridges. Myosin head tilts, pulls actin, so sarcomere shortens. So this was this part of the question. Now we come on to the last part of the question. Scientists compared the diameter of samples of striated muscle fibers taken from young mice and adult mice. The results are shown. The young mice is this one, which I'm coloring in yellow, and the adult mice are these ones, which I'm coloring in red. And you have muscle fiber diameter, and you have number of muscle fiber. Now whenever there's a graph, please look at the graph very carefully and then read the question and then come back to the graph. With reference to figure 7.1, describe 2 differences between the muscle fibers of young mice and adult mice, and it suggests how these differences may affect the sliding filament model. So with reference to figure 7.1, describe 2 differences. This is everybody can do that. So you got your 2 marks out of that. And then suggest how these may affect. So now coming back to this, we look at the graph. And when we look at the graph, then we see a lot of things which you can say, okay, so young mice have more muscle fibers that are smaller in diameter. Young mice is from here to here. This is the diameter, so they're between 10 and 48. So young mice have more muscle fibers that are smaller in diameter. Young mice have more muscle fibers that are smaller in diameter. Young mice have a smaller range of diameters of muscle fibers. Then you could give me a comparative data code. What is a data code? Young mice 35 at 30 and 16 to 44 is the spread of diameters, while the adult mice is 16 at 50. So it is 16 at 50. Young mice, number of muscle fibers at mean diameter in micrometers. So the at you look at 50, then you can see at 50 here what is happening. So at 35 at 30 in young mice and 16 at 50 in the 35 at 30. So it's talking about 30 here, and then it's talking about 15 here for the adult. So 35 at 50 and 16 at 50 for the adult mice. Spread of diameters, the young mice is 16 to 44 and the adult mice is 20 to 80. So you get the spread of the diameters. Then you have to give me the explanation. So in young mice, there are fewer muscle fibers or fewer sarcomeres. So less muscle protein, actin and myosin. So weaker contractions. So you could have given me those points and that would have got you your describe and explain. So young mice, fewer muscle fibers or fewer sarcomeres, or you could say the other way around, adult mice have more. Less muscle proteins, less actin, myosin, so weaker contractions. So this would all be the explain that young mice, YM is young mice and AM is adult mice. So young mice have more muscle fibers that are smaller in diameter and young mice have smaller range of diameters. Then you could have given me the number of muscle fibers in young mice, they're 35 at 30, adult mice 16 at 60, and spread of diameter 16 to 44 in young mice. This is about young mice and this is adult mice. So YM is young mice and adult mice. Now this is all the describe. But then it also said describe and then suggest how these distances may affect the sliding filament theory. So this is fewer muscle fibers, less actin and myosin and weaker contractions. So this would give you their explanation of that and that would complete your total number of 8 marks. Now I complete this video here and I'll continue the rest of the questions. We'll finish question number 7. So we left with 8, 9 and 10. We'll continue in third video. So we've completed question 4, 5, 6 and 7 in this video. The rest will go into the third video.